So welcome, everyone. The American Society of Cytopathology's Research and Current Concepts Committee is proud to present part one of Meet the Winners. This will be a 3-part video series that will help all of us to get to know and be inspired by the award winners from our 2020 annual scientific meeting. My name is Michiya Nishino from the Department of Pathology at Beth Israel Deaconess Medical Center, and I'm joined here by my good friend and colleague, Wang, from the Department of Pathology at Yale School of Medicine. And both of us are thrilled to meet today with 2 of the award winners from our annual meeting. We have Donna Russell from the Department of Pathology at the University of Rochester Medical Center, and the winner of this year's Cytotechnologist Scientific Presentation Award for her poster reporting their institutional experience with serous effusions. And we're also delighted to have with us Swikrity Upadhyay Baskota, currently a cytopathology fellow at the University of Pittsburgh Medical Center, and our first recipient of the Bernard Naylor Cytomorphology Award for her study characterizing the Peritheliomatous pattern as a cytomorphologic clue for recognizing melanoma in FNA samples. So thank you both for joining us today. So why don't we start with Swikrity? Can you start with a synopsis of your study?
Sure. Thank you, Dr. Nishino, for this opportunity, and thank you, everyone. First, the Peritheliomatous pattern in our study, we defined it as a sheath of viable tumor cells surrounding a central gaping blood vessels. We were seeing this peritheliomatous pattern in our daily practice at CD Site Cytology department whenever we were performing a FNA of a melanoma sample. And when we looked back in the literature, the peritheliomatous pattern was reported in various forms of melanoma in histology, but we did not find any literature reporting the peritheliomatous pattern in cytology characterizing melanoma. That's why we decided to do the study and compare the occurrence of the peritheliomatous pattern with the smears as well as the cell block. And it was a very interesting study. We decided to include the standard histology sections as well. So we compared the finding of the peritheliomatous pattern in the smears versus cell block versus histology. And for the cytology preparations like smears and cell block, we even quantitatively characterized the peritheliomatous pattern into low and high. If we were getting the peritheliomatous pattern more than 2 times in any of the specimen, we counted them as PP high or 2+. And if it was encountered very less frequently, like only once or twice, we counted it as low PP. And among the most preparations, we saw peritheliomatous pattern predominantly in histology, but cell block was more predominant than the smears while talking about the cytology preparations. So whenever we do not have the other clue like melanin pigment, as you all might know, melanin pigment is often not present. It's present in up to 40% in the cases that has been reported in the literature. Whenever there's absence of melanin pigment and the other features which are helpful might be the peritheliomatous pattern. If there is already a melanin pigment, we often do not go looking for other features, but the binucleation, pseudo-inclusions, cytoplasmic vacuoles along with the peritheliomatous pattern is helpful. It will help us order the melanoma markers so that we can save the sample and decrease our workload for more immunohistochemical stains panel, and we can save material for the molecular or other ancillary studies which will help in the personalized genomics treatment. Thank you.
Very good. Swikrity, this exciting study. So what are the challenges facing you when do this project? I understand a lot of work.
Yeah, it was definitely a lot of work, but I should thank my mentor, Dr. Pantanovich. He's always a very good guidance to work in any project as a trainee. Whenever I would go to him with any questions, like first few challenges that we faced was like we started including the fluid cytology specimens as well, but most of them had very marginal cellularity, and we were not able to assess whether they would present as peritheliomatous pattern or not. And technically it was not also fine needle aspiration, So he always had a definitive idea of how to move forward. With those kind of cases, we decided to exclude the effusion cytology cases positive for melanoma. So I would, and then other challenges we face is like, I as a trainee rotate through 3 different hospitals, and most of the cases we included was of UPMC Sadie side. But again, thank to Dr. Pantanavich and his coordinator, who managed to send me all the slides wherever I wanted. And then we sat together under the microscope and we discussed and agreed on each peritheliomatous pattern finding.
Yeah, terrific. Could you also just talk about what you found to be the most exciting or rewarding aspect of putting this project together?
Well, honestly, I encountered peritheliomatous pattern when I was signing out with Dr. Pantanavich, and it was his idea. But we were appreciating peritheliomatous pattern only in the cell block specimens. I had never given in-depth thought of looking for a peritheliomatous pattern, especially in the smears preparation. Then this study was more interesting when I started pulling all the slides and started looking for the peritheliomatous pattern in the smears as well.
So what do you learn from this experience and some advice to your peers?
Well, we as a trainee, we tend to involve in projects where we want to correlate with molecular or at cutting edge technology. But I felt like this experience of dealing with the different morphological features increased my in-depth knowledge of what I can find in melanoma specimens, which I found really helpful in daily life. Still, when I get melanoma cases, I look for peritheliomatous pattern, and it has been helpful for me in my daily practice as well.
Fantastic. Detailed cytomorphologic studies like this one, are really the bedrock, for our field. And, I was wondering if you can comment on future directions that you might have, about, this finding.
Well, we tried comparing the occurrence of the peritheliomatous pattern with any hotspot mutation, but we did not find statistically significant finding in our study. We believe that is due to a small number of the cases that we included in this study. If we do this study in a very large cohort, probably we can find, a specific hotspot mutation associated with melanoma predisposing to more peritheliomatous pattern than other.
Very good. So thank you so much.
Sure.
So I think we can move on to Donna. Donna is the awardee of the Cytotechnologist scientific presentation of this year, and do a very exciting study on tumor in peritoneal, pericardial, and pleural fluid. Donna, can you give us a highlight preview of what you did?
I sure can. Can everyone hear me okay?
Yes.
Okay. Malignant serous effusions are a common clinical problem in patients with neoplastic diseases. We all know that in cytopathology. And we know that cytologic evaluation of these effusions, especially because they're fresh, it's minimally invasive and an inexpensive procedure for the most part for cancer diagnosis, staging, and prognosis. So we wanted to look at, we wanted to analyze both pleural, pericardial, and peritoneal effusions in this study, and to try to find the primary site of origin. Of course, there were those sites, where the tumor was of undetermined origin. But for the most part, we had great cell block material on all of our cases included in the study. And we performed immunochemical stains and molecular testing on those for tumor profiling. But the overall aim of this study was to determine the prevalence of malignancy in both the male and female populations in all types of specimens, because the literature sort of had a void from current literature anyways. So we looked at confirmed malignant cases in all 3 categories. These were identified in our electronic database here at the University of Rochester Medical Center. We also looked at clinical history and immunohistochemical results. They were all noted and correlated in this study. Over this period of time of 5 years, we found 2,540 serous fluid specimens that were received within the laboratory. And the most common type that we received were pleural effusions in both the male and the female populations. 560 cases, or about 22% of our serous effusions, were positive. We also looked at age ranges, which, it ranged from anywhere from 5 to 98 years of age, or 97 years of age in both the male and female categories. Females sort of had a slight preponderance, in the effusion, all effusion categories, 1.3 to 1, so just very slight. And the most common type of malignancy that we did find was adenocarcinoma in this study. In men, adenocarcinoma of the lung was followed by GI, which is a common finding. So we didn't really see any shifts in paradigm there. But in women, lung adenocarcinoma in our study was more prevalent, followed by breast carcinoma as a cause of malignancy. So we found this slight shift in paradigm in the female population, with lung adenocarcinoma being the most common type of malignancy. And this is a great study, I think, because effusion cytology is fresh. It's the ideal specimen for ancillary testing for both IHC and molecular testing. That's about a synopsis of the study.
Thank you. So,
go ahead.
What really kind of excites you to start this project?
Well, I love working with the residents and fellows in our institution. I'm in charge of the residency cytopathology and fellowship cytopathology program here. I also work as a program director at Roswell Park Comprehensive Cancer Center, and we have 3 students this year, cytotechnology students. So I love to teach, and research is a big part of that. And I love all aspects, from data collection to taking images for the poster or papers. I just love working with people as a team and trying to help residents and fellows and students just get excited about cytopathology. It's a passion of mine. It always has been.
Thank you so much. Just to add to that, when I was trained at University of Michigan, the first people show me slide are cytotechnologists. I'm really grateful. It's very, very rewarding experience.
Yeah, your role as a mentor in this project, for trainees is much appreciated as well. Now, can you talk to us about any challenges that you faced, during the project, and how you went about tackling those?
Well, it was during the pandemic, so it was a little difficult because at first our residents were not, in-house. We were not. Right. We were doing everything by Zoom. So data entry, even amongst the cytotechnologists that were involved in this project, we had to use a shared site to get some of the data entry into. So those are the challenges here. And then, of course, just getting everything together. I think there were so many versions that we had to email back and forth. It was hard to keep track of all those versions for the final version that we were going to submit.
What kind of experience you want to advise to your peers learning from this project?
Well, I think I find value in teamwork. I think that, when you work together as a team, you can accomplish anything. So in this study, our resident Phoenix Bell who participated. Our at that time she was our resident. Now she's a fellow. Our cytopathologist who was involved, Dr. Tanupriya Agrawal, and my co-workers, two cytotechnologists who I work really well with, Roshana Lynch and Ryan Granger. We all work together, and I really think that you can accomplish anything. But also the fact that this data pool is so large, it's a 5-year retrospective study. I'm looking forward to multiple future studies from this data. I mean, we could look at IHC stains for specific entities, and we have used this database before for other new and upcoming stains and other projects.
Fantastic. Institutional experiences, like the one that you brought together, serve as a really important point of comparison with our own practice patterns, and also useful for informing meta-analyses and standardized reporting systems. And your study is so timely, in light of the international system for reporting serous cytology. And the journal of the ASC is always looking for terrific studies like this one. And can you tell us about your plans for publishing this?
Well, my motivation for this project was largely due to my participation as a chapter author from the international system for reporting serous fluid cytology. The editors, as you know, are Dr. Crothers, Barbara Crothers, Dr. Asha Chandra, Dr. Daniel Curtis, and Dr. Fernando Schmidt. It truly was an international effort. But Dr. Crothers and I were discussing, gaps in the literature. So at a recent CAP meeting, and I just decided I was going to come back and try to make sure this could get into the literature. So definitely, of course, it's going to be published in our journal, JASC, as soon as I find time to get it written up. But it's in the process. It needs a little work.
That's have to say there'll be a lot more versions back and forth, right? Much more back and forth, more and more writing. Yes,
especially since our resident is in Boston now, and so we're definitely going to have to do a few drafts of the manuscripts. But we work well that way. We figured it out, I think, during this pandemic.
Very good, very good. Thank you, Swikrity and Donna. So again, I thank you for this really excellent work, and congratulations on your work again. As you know, cytopathology is still a very young subspecialty compared to other specialty of pathology. So there are much more room, a lot more room to prepare and standardize the preparation and immunostain and salivary test. So, and there are also a lot of room to characterize the morphology of a lot of different tumor types. So the Bernard Naylor Award really opened the avenue for this in-depth understanding of the morphology. I encourage new generation to go to this direction. And for our Cytotechnologist Science Award, our Cytotechnologist is really kind of middle level health providers. So now we have the master degree, and we really want to encourage more of our cytotechnology to do science and to teach our young residents. So by this two award, we try to encourage more of this kind of effort, and really hope that we can build a stronger cytopathology subspecialty. So thank you again. And really, we hope next year we'll have more applicants for this both awards. Thanks.
Thank you. Thank the ASC.
Thank you. Thank you, everyone.
Congratulations.
Thank you.
Thank you.
Welcome to part 2, meeting the winners presented by the Research and Current Concepts Committee. This will be a 3-part series of all the winners from the 2020 Annual Scientific Meeting. We are joined by Dr. Robert Post and Melissa McCrowson, and they'll be giving a brief summary of their excellent work. So first will be Dr. Post. The winner who won the Warren R. Lang, MD Resident Award. It's established in 1978. The award is presented annually to recognize a resident or fellow in an approved training program who submits the best scientific paper in cytology at a poster or platform session during the ASC annual meeting. So Dr. Post, if you could please give us a brief summary of your poster, that'd be wonderful. Thank you.
Sure. Good afternoon. I'm Dr. Robert Post. I'm a current third-year resident at University of Illinois. So my presentation, my poster, my research project was motivated behind this, the observation that in patients undergoing anal cancer screening, HIV infection is a significant comorbidity. So we were interested in clarifying the correlation of HIV infection status with anal cytopathologic diagnoses, and as well as HPV infection status. So at our institution, anal pap cytology specimens are processed using the ThinPrep system from Hologic, and HPV testing is done on the Roche Cobas 4800 system. So we have it identifies HPV genotype 1618 as well as a kind of a catch-all other high-risk HPV category, which is 12 other high-risk HPV genotypes. So in order to build our cohort, we queried Cerner PathNet case retrieval for all patients who underwent anal cytology and had concurrent HPV testing between November 2017 and March 2020. And we reviewed all the results, and we ended up with 159 cases that met the criteria. 31 patients out of this cohort of 159 were positive for HIV, and that was so it was about 19% of the identified cases. So in the poster, you can see Table 1 summarizes the distribution of the cytologic interpretation among the HIV negative and HIV positive groups. The distribution of the interpretations were similar between the two groups. One thing to note was that ASCH and HSIL diagnoses comprised 13% of HIV positive cases, but only 3.88% of HIV negative cases. However, due to the small cohort size, we did not find this to be statistically significant. In Figure 1, which is the top pie chart, we can see the distribution of high-risk HPV genotypes in the HIV negative group. And then, so you can see that HPV other and HPV other plus 1618 were comprised a majority. Figure 2 shows the distribution of high-risk HPV genotypes in the HIV positive groups. Just to summarize this, 84% of HIV positive patients tested positive for any of the high-risk HPV, compared to 72% of HIV negative patients. In the subgroup of patients with negative cytology, 83% of HIV positive patients tested positive for any high-risk HPV types, compared to 57% of HIV negative patients. And, because our cohort was small, this wasn't statistically significant. But we found that a majority of patients tested positive for high-risk HPV genotypes other than 16 and 18, regardless of HIV status. And was about 66% of the HIV positive group and 71% of the HIV negative group. In our cohort, the other high-risk HPV genotypes were far more prevalent than genotypes 16 and 18. So our conclusions were that high-risk HPV genotypes other than 16 and 18 are more prevalent regardless of HIV status, and especially for HIV positive patients. This finding we think that this finding could have implications in the management of cancer risk, as well as developing vaccine-based prevention strategies. So
Awesome.
that's summary.
Great, thank you. That's wonderful summary. Thank you so much. So what motivated you working on this project?
So I had an early interest in cytopathology. So I started doing, I started with a rotation in my second year. And at the time, the current fellow was working on a project examining anal cytology and HPV status. So I thought that his was a really interesting project, and I worked with him to try to develop an offshoot project. And so one thing that we had discussed when we were discussing his project was how many of these patients were HIV positive. So the two of us working together developed as well as with the attending too, developed a project to look at how HIV correlated with the cytology and HPV infection status.
And so what did you find most exciting and rewarding about working on this poster and project?
So beginning this project, and with a literature review and realizing there wasn't much known, there wasn't too much literature on this. So doing a project where not much is known is exciting. I find that very rewarding and exciting. I find that the idea that research could contribute to future guidelines or management of cancer risk, or development of vaccination guidelines, even if it's just a framework or beginning of a foundation for more research in the future, that's very rewarding to me.
Thank you. And so as a trainee, what were your biggest challenges? What were your challenges as a trainee?
Yeah. I'm not sure if this is a trainee specifically, but I mean, one of the biggest challenges of doing this project was just because this was an outreach patient population, the patients often had like a very incomplete medical history. And it was hard to know whether sometimes it would say that, or it would not suggest that the patient was HIV positive at all, but perhaps they were HIV positive. We had no way of knowing. And perhaps that was a challenge as a trainee, because if maybe I would have been able to, felt more comfortable reaching out and finding out more information about these patients, or I don't know. I found it difficult as a trainee because I felt like that was what I had to work with. So I had to try to make sense of the data as I had it. And it ended up with some fairly noisy data as a result, where I suspect that some of the patients who were in the HIV negative group were indeed HIV positive. But I have no way to show that.
Interesting. Thank you. That's a great point about only be able to work with what you have.
Yeah.
And I think you kind of dovetailing into your last your last point, what did you learn from this experience, and how would you advise others who are trying to do similar projects in the future?
Yeah. So I learned the importance of and difficulty of collecting good data, particularly from medical records, and particularly from medical records from. I mean, medical records can have various levels of, I guess, accuracy or reflection on what's going on. So I would advise other residents or trainees interested in doing research to start early and exercise a lot of patience with the data, and just give time to think about it, and give time, and additionally discuss the data with the other members involved with the project, as well as other faculty members who are familiar or involved, because having multiple sets of eyes on the data can be really helpful in uncovering trends and just finding some meaning in the data.
Great, thank you. Have you submitted this as a manuscript to the journal, or?
I have not, but I mean, I think in order to do that, this project would require a bit more work. Maybe building a larger cohort size, or ensuring that the data is a little bit more reflective of the patient's HIV status or whatnot. So I am potentially considering doing that with the current fellow. But I also have some other ideas in cytopathology research. Then I would be happy to submit a manuscript to JASC.
Oh, yeah, well, excellent. I look forward to reading that if and when you do. That'd be great. Thank you, Dr. Post, so much for sharing your really interesting work. And thank you. We really appreciate your time.
Thank you very much.
So we will move on to Melissa McCrossan.
Yes.
Thank you so much for joining us.
Thank you for having me.
Do you mind sharing her poster? Thank you, Jamie. So as I said, we were joined today now with we're joined by Melissa McCrossan, and she won the award for Innovative Cytotechnologist Practice, established in 2018. This award demonstrates programs, services, or projects performed by a cytotechnologist that are creative, cost-effective, and improve the practice of cytopathology. And so if you wouldn't mind, please sharing a little bit about your work. And thank you.
Thank you, James. My name is Melissa McCrossan. I'm a staff cytotechnologist and the specialist technologist of education at Northwell Health in the cytopathology laboratory. So our poster, Training in Cytopathology in Times of Social Distancing, a comparison of remote versus traditional learning, is a summation of our lab's experience with the COVID-19 pandemic and how we modified our cytology program to remote learning to continue to provide the necessary training for our residents and fellow. For our cytology program, we took known study set boxes and multiple unknown slide test boxes and scanned them into our eSlide manager. We already had online study exams created a few years ago by myself, and we had slowly been introducing them into both our residency and fellowship programs. And we also found and utilized old ASCP testing materials that were on CD-ROM, and we moved them to an online format and created worksheets that went along with the online work. And our health system instituted Microsoft Teams and OneDrive to communicate between departments and to share materials with each other. So we created policies and a schedule breakdown for our residents and fellow to follow and keep them on track while being off-site. We had daily morning lectures. Our residents researched questions given to them by their assigned attendings. They screened the unknown and known cases by accessing the eSlide manager from their home computers. We had daily sign out and they worked with their attendings via telecytology, and they completed the daily workbook assignments. We also provided numerous websites to access videos of FNA techniques and others that simulated slide screening. In lieu of in-person ROSE FNAs, we created a simulated ROSE FNA experience through the Teams app and telecytology. We used known cases and simulated to our residents and fellow what it would be like to perform ROSE over telecytology. At the end of each rotation, we gave each resident and fellow an evaluation form to fill out. We also gathered their grades from entry and exit slide screening exams, online exams, and the daily work assignments. We found that most of our residents and fellow performed at the same level as they did prior to the COVID-19 pandemic. And we also had a few of our trainees that performed exceptionally well. Gathering the data from the evaluation forms, we also found that the majority of the residents and fellow felt that the amount of work received was comparable to that pre-remote learning, and that they felt they learned about the same to more as previous rotations, and that they wanted to continue to receive their work virtually. They found that the workload was easier to manage with having the option to work remotely or in person, And we found that to overcome the lack of hands-on experience of performing ultrasound-guided FNAs due to the closure of our clinic, that our attendings gave didactic lectures on the basics of ultrasound-guided FNA performance and interpretation of ultrasound-guided images. In conclusion, we found that remote learning performed just as well as in-person training, and that by combining the two, we can create a stronger and more robust program that is prepared if we are faced with any unprecedented challenges in the future.
Excellent. Thank you so much. So as a person who also, as a cytotech who also works at a academic institution, it's been a challenge, you know, and this is a really timely presentation. So I really enjoyed going over it before this session. So what motivated you? I mean, obviously we know what motivated you to work on this project, but you know, what you know, specifically, what did you find most you know, most motivating about this?
Well, I definitely was motivated because it was decided that our residents and fellow were now working from home, and it was like, okay. And I had to make sure that our program continued going and that it would still receive the same quality of education online as we did in person. So I needed to think outside the box and find creative ways to mimic our residents' daily workflow at home as it was in the office. I definitely pulled from past experiences from my own training way back when in cytology school, where I just thought of how my professor had combined, I'm dating myself, Kodachromes with true slides and online. So I use that as a guide to think outside of the box and how can I do that for my residents, and to make sure that we were still up to the standards needed for our residents to pass their boards and increase our patient care. I then decided to submit it to the ASC because I wanted to share our experience and also hear about other labs' experiences and how either they were similar or different.
Excellent. Thank you. So kind of dovetailing off of that, you know, what was the most rewarding or exciting part of this project?
It was definitely exciting and challenging to have to recreate an entire program in such a short amount of time without losing the integrity of the program. And none of us knew how long we would be remote teaching. But now retrospectively, looking back at it, I found that the challenges were quite inspiring, that our residents unexpectedly thrived in a new learning environment and adapted quickly and developed new skill sets. Seeing that our residents stayed the same and some even performed better definitely provided comfort to me, knowing that I was still providing the same level, if not a better quality of education. And due to this, we now successfully incorporated new training methods such as telecytology into our program, which we had spoken about but hadn't really incorporated fully until now. And we're finding that it could hopefully lead to an advantage for our residents and fellow and when applying for fellowships and faculty positions.
Oh, great, thank you. So as a cytotechnologist, so I mean, I know that your role is in education, you know, is the educator for your department, but were there any special challenges? I mean, obviously, other than, you know, building an entire training program from scratch, which is amazing, or redefining your training program. What were there any special challenges as a cytotech? You know.
Yeah, definitely one of the biggest was trying to maintain my own workload as a cytotech, because obviously patients always come first. So that was definitely trying to maintain the workload while developing a program. I also was working with multiple faculty and fellow coworkers to coordinate training modules and making sure that everyone was following on the same page, and that all aspects of the residency training were covered, because our own fac was affected by COVID-19, and so we had some shortages here and there. We also encountered computer issues when accessing eSlide Manager, connection issues with the computer, lack of communication or lost communication during emails and conferences. And then our biggest challenge was that we didn't have the hands-on training for FNA rows and for microscopic screening, which are invaluable skill sets that can only be developed with fully hands-on, and not being able to do that was like the biggest challenge.
Mhm. Yeah, I can totally see those as being extremely hard to work out. So how would you advise others, in your experience, building this? Obviously we're going to be in this for a long time. So how are what experiences would you share with others?
I would definitely say that it's amazing and what you can learn when facing adversity. It definitely forced us to reevaluate our past policies and procedures and implement new policies and procedures, especially regarding to items that were discussed in meetings, but we never had the urgency to pursue further. That now we look at everything differently, is in terms of, do we need to do this now and implement this now, or is it something that could be on the back burner? It definitely made us think outside the box, push outside of all of our comfort zones in terms of telecytology and remote teaching, online screening of full slides that not many of us are experienced with or comfortable with. But, you have to do what you need to do to make sure that your residents and fellows successfully meet the requirements for graduation and have success in their careers. And I'd also advise people to not be so hard on themselves, that everything's a work in progress and is constantly changing, and we need to adapt with the change.
Excellent, thank you. And also, are you submitting this to the JASC?
It's a work in progress, yes, I'm hoping.
Yeah, definitely. Well, Melissa, thank you so much for joining us. I really appreciate your time. And Dr. Post also, thank you so much for your time and your excellent work. These are really excellent presentations and things that are really important to think about. So again, we really, we appreciate it. Thanks.
Thank you so much for having us.
Thank you.
So welcome to part 3, Meet the Winners, presented by the Research and Current Concepts Committee. This will be a 3-part series of all the winners from the 2020 Annual Scientific Meeting. I'm Diana Ng. I'm a cytopathologist at Memorial Sloan Kettering.
I'm Diana Lynn. I'm a pathologist at the University of Alabama at Birmingham. So we have the Quality Improvement in Cytology Award. Established in 2019, this award recognizes quality improvement projects designed to improve quality in cytopathology practice. The award will be given to those projects that demonstrate innovative and forward-thinking approaches to current cytology practices, as well as measurable and replicable improvement. And this award went to poster 107, Quality Data Abstraction Utilizing Bethesda Pap Testing Reporting, Key Implementation Science Indicators for National Gynecologic Cytology Benchmarks. We also have the Advances in Thyroid Cytology Award. Established in 2014, this award recognizes the abstract presentation that best contributes to the knowledge and diagnosis and treatment of thyroid diseases using fine needle aspiration and/or ancillary techniques. This award is supported by an educational grant from Thyroid Cytopathology Partners in Austin, Texas. This went to Platform 9, Immune Profiling of Thyroid Lesions and its Application to Malignancy Prediction.
Okay, Amy, maybe you can introduce yourself.
Hi, I'm Amy Spitzka. I serve as the Senior Director for Quality and Patient Safety with the American Society for Clinical Pathology. By way of training, I'm a laboratory professional in histopathology, cytopathology, and also molecular biology.
All right, and I'm George Schur. I'm a graduate student at Vanderbilt University in the Department of Molecular Pathology and Immunology.
Okay, well, first off, congratulations, Amy and George, for your awards. I think this is a great honor. So We can start off with just having you guys give us a summary presentation of your work.
So thank you. Also want to recognize the American Society of Cytopathology for this recognition, as well as the opportunity certainly to share the work that we've put forward. As you can see in our presentation, we focused on quality data abstraction, really looking at the Bethesda system PAP test reporting to identify key implementation science indicators that could be potentially utilized nationwide for GYN cytology benchmarks. So by way of aims, we started out looking at mapping clinical quality measures, this long-term to assure benchmarking indicators, again, aligned with PAP test reporting. And what we did within the infrastructure of this, given our National Pathology Quality Registry, is we assured that the Bethesda reporting, we used the 2015 version, aligned with PAP test quality measures. And from that crosswalk, what we were able to deduce is that continuous quality improvement benchmarking indicators can be readily available across unique features of the Bethesda system, and really make sure that there's a conglomerate of meaningful data that can be put into individualized quality measure type data. So just quickly, the National Pathology Quality Registry really measures for performance improvement to elevate patient outcomes. And we're also a CMS qualified clinical data registry. We span all laboratory specialties, including cytopathology. And when we think about guidelines and measures, we're driven by data as a national quality measurement platform. So while we can look across all of the specialties within the lab, for instance, monitoring appropriate utilization, improving pre-analytical processes, even considering optimizing turnaround time, critical value reporting. Critical to cytopathology is that we can also assess analytical and diagnostic accuracy. This helps us establish best practices, through national and peer group comparisons. I bring this to light because as we recognize the relevancy of the Bethesda system, we wanted to apply it in the domain of continuous quality improvement. Additionally, I think in fairness to the real life experience that so many of us share, is that as we're implementing or optimizing our laboratory information systems and EHRs, there's really an opportunity to take a highly structured reporting system and identify how it is we can decrease administrative burden, but assure safety for our patients just based on this clinical quality measure set. I'll stop there. That's really an overview of what our project is.
Well, thank you, Amy. I think that was great. And I'm really excited that you're using implementation science frameworks. As a burgeoning implementation scientist who's really interested in uptake and also monitoring evaluation, I'm really excited that you're using implementation science. So I guess we'll just move on to George in your presentation.
All right, so for my project, my goal was to investigate genetic and immune predictors of thyroid lesion malignancy and outcome. We found that computational methods that use RNA sequencing data are capable of revealing distinct immune infiltrate profiles in different subtypes of thyroid lesions. Using this data, we've aimed to test whether immune infiltrate data can improve thyroid malignancy prediction. We've also tested the ability of this computationally derived data to predict things such as advanced thyroid tumor stage and poor outcome. Our preliminary results suggest that RAS mutation, in combination with computationally deconvoluted levels of infiltrating B cells, CD4 T cells, macrophages, and neutrophils, improves malignancy prediction, even in thyroid lesions without a BRAF TP53 or TERT promoter mutation. Additionally, we found that macrophage levels may be used to improve tumor stage prediction. And finally, we found that TERT promoter mutation, TP53 mutation, and macrophage levels each may predict future poor outcome.
So thank you, George. That was a great summary. I was at your platform moderating, so I know how in-depth and how interesting your data are. I think the role of kind of tumor infiltrating lymphocytes and kind of immune response in thyroid cancer has really been underexplored. So I think this is really exciting work. Thank you. So since I asked question 1, maybe Diana can ask question 2.
Sure. So what motivated you to work on this project?
So really the motivation behind this project is on several levels very practical. Specific to proof of concept, we wanted to drive continuous quality improvement on our patients' behalf. So first and foremost, certainly being patient-centric. On the very practical side of this, as well as I have to say being curious, we recognize fully the relevancy of the Bethesda system when it comes to Pap test reporting. We also recognize that the Bethesda system has really integrated well to clinical practice. And when you think about permanence in practice and the consistent utilization, it was a safe model to base curiosity of implementation science on. And the reason I say this is when you think of data points spanning the entire laboratory, clinical laboratory discrete results are clearly that much more not only readily available, but easy to crosswalk by way of clinical quality measures. It's discrete data, it's very specific, it's a range, it's not necessarily descriptive as we've come to know in pathology. But the Bethesda really offers, again, that relevancy and permanence to apply some concepts for crosswalking and algorithmic type building out of what this can mean for clinical quality measures. Additionally, I have to share that, there are so many of us. I historically was in the role of education leadership with Mayo Clinic. I also served as anatomic pathology operations manager with Mayo Clinic. And, for many of us in those roles or similar roles, even pathologists, there's extensive administrative burden. A lot of work goes into what we do to prove our level of quality. And that's well worth, I think there's a strong return on investment. However, I was curious, as was our team, as to what we could do to drive down that administrative burden while we recognize and reconciled, again, the benefits of Bethesda systems. So really it comes down to healthcare data mapping to clinical quality measures and how we can identify benchmarking indicators for continuous quality improvement. And in essence, we developed an algorithmic crosswalk that designated our Bethesda system GYN cytopathology to quality data points. So it was curiosity, but it was also the need to be incredibly practical and to give back to the community while assuring our patient safety.
Actually, Amy, I don't think everyone knows what implementation science is since it's a relatively new field. Maybe you can just give a brief definition.
Sure, I'll certainly strive to, meet on all the impacts of it, but please feel free if there's something that I forget. I think implementation science, at least from my perspective, where I work now as a leader for quality and patient safety, really relates to continuous quality improvement, but it includes those human attributes as relates to even data points that we can qualify and quantify, such that as we go forward with implementing, it can be products, it can be services, but it can also be quality and assuring quality. It's that the science that assures each step of an iterative process is confirmed and articulated, documented, such that that is known moving forward. And in this case, it's really building out an algorithm. Would you like to speak more to it? I'm sorry.
No, I think just my soundbite would be, I think the way I see it is the field that allows us to bridge evidence to practice gaps.
Yes.
That's how I usually define it. Just kind of how do you, for example, we all know that brushing your teeth is really good for you, but how do you get everyone to brush your teeth reliably?
Yeah, and I think it's that evidence to practice in the framework of this is, continuous quality improvement. But again, the practice of the Bethesda system, right? On every pap test report, you know, we must use it. It's, to your good point, brushing your teeth. But because there is such permanence in practice, implementation science works well in this regard, building a bridge from experience to reliable practice. Thank you.
George?
Okay. So the question was, what motivated you to work on this project, right? So one of our main motivations for this project was to better understand markers of aggressive thyroid cancer. The ability to predict thyroid cancer malignancy has really greatly advanced over the recent years. But a question that hasn't been as thoroughly explored is whether or not we can predict which well-differentiated tumors are most at risk for future disease recurrence or aggression. So I was interested in learning whether some emerging tools, such as immune infiltrate deconvolution using RNA-seq data, might provide a way to anticipate outcomes and guide treatments.
Yeah, and I think your work is very critical, just because molecular, as great as it is, there hasn't been an algorithm that predicts aggression and prognosis. I think the molecular data are really great in really stratifying how malignant it will be, but I think there really needs to be a lot of work done in terms of stratifying prognosis. So next question for both of you would be, what did you find most exciting and rewarding about this project?
Thank you. I think really the reward came from the unknown. We entered into this hoping to discover how we might align HL7 and data points to the Bethesda system reporting. So we used HL7 standards. It was version 2.8, and we transferred Bethesda system patient data. We were very strict in our criteria, both for PHI and HIPAA standards. And as you can well imagine, you know, the two indicators within pap test reports, so specimen adequacy and then interpretation or diagnostic reporting parameters, were abstracted. And I think, in fairness to the practice of the Bethesda system, there's definitely the published strict reporting criteria. And so we drove to that first in our initial algorithm. This again to identify a crosswalk of numeric indicators mapped to clinical quality measures. So when we think of mapping and we think of numeric indicators, again, it was a little bit of a entry point for us by way of looking at specimen adequacy. We could really look at binomial numeric indicators, satisfactory, unsatisfactory, and then look at unique indicators for all of the Bethesda diagnostic categories. Again, starting with very strict reporting criteria, but we wanted to be respective of reporting practice nuances. The reason I share this is if we were to keep our algorithm only with strict criteria, we recognize across all of the practices and even patient nuances that our quality indicators would not be as viable or as valid, given the fact that some of the reports would fall off. So we started with strict criteria, and then our next iteration really looked at assessing potentially divergent pap test reporting indicators. We wanted to coalesce, make sure that we had good convergence around identifying any reporting indicators that would link to the Bethesda system. So at that point, we had two broad constructs. Certainly the specimen adequacy was the easiest and the most secure. And then what we did is we reviewed patient pap test reports. And as you're probably aware, HL7 falls into OBR and OBX segments, specifically observation requests and observation results. And those segments and sequences, really in their entirety, can be abstracted for linkage to the assessment to the numeric indicators algorithm. So, the excitement, the intrigue really came that there were so many unknowns, but again, a belief that if we approach this from, again, more of a learning interface and implementation science, what we could do is holistically map strict Bethesda, potential nuances, and then ultimately integrate optimal CQM mapping. And that, I think, was, you know, if we can boil it down into one outcome, that was incredibly rewarding. That's it.
Right. So to answer your question, what did you find most exciting and rewarding about this project? One of the main reasons I'm really excited about this project is because of what I think is its potential impact. This project uses clinical specimens we collect at Vanderbilt University, and the findings we think have the potential to directly change clinical care and improve patient outcomes. Additionally, I'm excited for this project since it helps me to build my computational skill set and advance my career. So as a graduate student, it's really important for me to be learning and keeping up to date with methods that are useful for my field of research. So I've really picked up a lot of skills in coding, like in Unix and R, and like a lot of other like platforms that I've had zero experience prior to starting this project.
Okay, thank you. So I'll move on. What were the challenges you faced in completing the project, and how did you address them?
I think the challenges that we experienced were along several different unique experiences. Certainly technology, you know, interfaces, feeds, scrubbing data, all of that. Certainly look to our data partner as relates to not only assuring patient safety, but also making sure that we had valid data. So that had to be a baseline, and one that, you know, was expertise in the field we were able to achieve. I think in fairness as well, looking in past experiences of my career, the Bethesda system is a language, right? It's a language that feels very at one for us. It's a level of comfort. Even though I'm not currently a practicing cytotechnologist, you know, I can envision the days of being at the microscope and really having that reliability of that, again, viable language. So too for our colleagues in information management, you know, when we speak of languages such as HL7 and clinical quality measures, OBR, OBX, those are languages that they gravitate to, add meaning, and characterize, in our case, true quality on behalf of our patients. So it was really the ability to not only explore new language skills. You know, we've been learning this as a community, as a scientific community throughout, again, looking back at LIS and EHR implementation and optimizations. If we lean into these languages, what we find is that we can have truly data-driven quality measures. So overcoming that challenge, making sure that the Bethesda system as a taxonomy could again match to CQM logic, really help to build an algorithm linking Bethesda to HL7. And I think, you know, that capacity and the validity to really align quality indicators is shown in the data. We had over 1,300 aggregated PAP test reports. So we overcame the technology challenge. Those were inclusive of adequacy and diagnostic indicators. And then, as I shared with you, both the initial iteration and the more nuanced iteration of Bethesda yielded over 6,400 unique PAP test results. We confirmed all of that with Bethesda system reporting and truly found 100% accuracy of Bethesda system reporting aligning with that abstracted PAP test numeric indicators.
Right, so I'll go ahead and answer the question. So one challenge of my project was just managing the large and very complex set of clinical and sequencing data for our many patients and samples in our cohort. Another challenge of this project was learning how to set up the code to get the raw RNA sequencing data to a gene expression count table data that could be used for immune deconvolution. While I had some experience with next generation sequencing library building in the past, processing these results of the data was not something I knew how to do until I entered grad school. However, with the guidance of my professor and our collaborators, as well as some basic coding experiences I picked up in grad school class and in previous research, I was able to learn how to set up and run the code successfully.
Yeah, that's something I've noticed too with my own personal projects. It's just how to manage large amounts of data. I think both Amy and you, George, you're dealing with very large data sets. So how do you organize them and how do you analyze them in meaningful ways? I know your projects are very different, but clearly data management is a universal theme in a lot of research projects. So in terms of kind of moving forward with the questions, what are some future directions for your projects? What's your next step?
Thank you. When we look at future directions, to your good point, as relates to large data sets, manipulation, making sure, again, validity, and just confirming all data points, there are some real opportunities to engage with some, I would say, emerging technology to make these more scalable solutions. So suffice it to say, this was the creation of, again, indicators, the Bethesda reporting, an algorithm linked to, binomial and multinomial indicators that we could crosswalk. So future directions really, opening up dialogue with other clinical sites and even technology partners to make scalable solutions. When you think of this on a broad scale, to the benefit, again, of our patients, and even with what George had said, with identifying an algorithm that's meaningful even outside our initial look, we can link to measures, everything from rate of major and minor discrepancies, identifying total discrepancy rates in overall cases, even simple things such as rate of review of Pap test samples interpreted as negative, making that as a benchmarking indicator, site versus site, without a lot of the heavy lifting or administrative burden associated with that. So ultimately, towards benchmarking, we'd like to map clinical quality measures to assure patient safety and Pap test reporting. If we could get a view or vision on this by way of more national type standards, I think that would be meaningful to the community. And it wouldn't necessarily mean individual locations looking at what their reporting outcomes are. And from assuring that crosswalk with Bethesda system, we can even more assure aligned Pap test quality measures.
Yeah, and that's the cousin of implementation science, is dissemination.
Exactly.
So that's always the natural next step. So George, what are your future directions?
So for the next steps of my project, we're in the process of expanding our cohort, and we'll be receiving additional thyroid cancer samples from the University of Washington, from one of our collaborators, including anaplastic cancer samples, which are pretty rare and valuable for our means. But this will greatly improve our ability to study aggressive thyroid cancers. Additionally, once we have our additional samples, we will be able to have the statistical power to evaluate combined models, such as the model looking at TERT promoter mutation, TP53 mutation, and macrophage levels to predict future poor outcome altogether. We also have plans to compare metastases to primary tumors of the same patient, so sequential samples. And finally, we also plan to evaluate a larger cohort of fine needle aspiration samples using flow cytometry. So our initial flow cytometry of about 32 fine needle aspiration samples looks really good and supports our findings so far. But we plan to collect additional samples to prove the ability to detect these alterations with flow and not just RNA sequencing.
And another question, so what do you envision if this became a clinical test? How would you envision this actually kind of realizing itself? Would this be like a multiplex PCR where you have a small panel kind of whittled down from the RNA-seq data? Or do you feel like this would be a flow-based test?
So I think, we have RNA sequencing data and then we have flow. And I think the benefit of, say, on one side with RNA sequencing is that patients already get a lot of genetic testing of thyroids, RNA-seq and DNA-seq. So since we already have that data available, it's not too much work to do additional analysis on that data. But I think having both is really important. like RNA sequencing and flow to like validate each other, and also existing tests that, you know, predict malignancy using genetic testing, I think are still very valuable, but looking at immune deconvolution is just adding another angle on top of that to give you more resolution and more detail.
I would love an automated multiplex PCR. If you could ever make that, I would love that.
I'll definitely look into it. Yeah, thank you.
Yeah, great. It's always good to hear that it's not just one project. You've got more stuff going. You're going to get more and more projects out of this, because you know, both of you have such great projects. You know, you don't want to stop there, just keep continuing. So the next question is, what did you learn from this experience that you would advise others?
I think one of the key takeaways from our experience is that the exploration of new solutions to required work really can help identify some new approaches, and again, those language skills, the convergence of technology with something that we've become, you know, that's really historical, foundational in our experience, such as the Bethesda, that the efficacy of technology can really elevate that. And I think the other recommendation that I would put forward is just the value of benchmarking to patient outcomes. So to the good point of, you know, dissemination as part of implementation science, it's broader than just, you know, a single institution, and serving certainly for patients nationwide. And this is an algorithm that could work by way of benchmarking and really supporting PAP test outcomes.
All right, so to answer your question, I think this experience taught me that organization, as we were discussing before, is really important when maintaining a large cohort of data. So by keeping, say, like multiple copies of all of my records as they're updated over time, I can always go back to ensure that I know what edits to the data are happening and when. The experience also taught me that data and findings can be surprising, and that science doesn't always work out the way we predict, which is why we call them hypotheses. For example, prior to our analysis, B cells were not really something that we had been thinking about much, but our data for B cells turned out to be really surprising and interesting across our thyroid lesions. We're still not sure why yet, but the data holds up and should give us more insight into thyroid cancer metastasis and progression.
Sure.
And congratulations, George.
Oh, sorry, I'm muted. Congratulations, Amy, as well.
Okay.
Yeah, and thanks again, everyone.
Sure.
Yes, thank you. This was a positive interaction. Appreciate you all.
It was great. It was okay.